Welcome to the Center for Youth Wellness's training on ACEs and toxic stress. In this module, we'll cover epigenetics. The purpose of this module is to give you a broad overview of what epigenetics is, the impact of ACEs and toxic stress on DNA and DNA expression, and give you an overview of what telomeres are. So what is epigenetics? Epigenetics is an emerging scientific field. Epigenetics can be thought of as the study of what's happening on top of, or over, or above our genetic code, our DNA. If we break down this term, epi means above or on top of, and genetics refers to our human genome. Every cell in the human body has the same DNA code, but every cell doesn't look or function in the same way. This is because the DNA sequence is read and translated differently for different types of cells. With epigenetics, we study the way that external factors can change this reading and translation of our DNA, which then determines cellular structure and function. External factors do not change the DNA sequence, but impact how genes are read by turning them on or off. One way to conceptualize epigenetics is to think about books stacked in a library, waiting to be read. Epigenetics is the likelihood that the books will be taken off the shelf, opened, read, and utilized. When you go to the library, you don't read all of the books that are there, just the ones that are useful to you. Epigenetic changes don't modify the DNA infrastructure or the library itself, but they can alter the expression of DNA, which books are read. This then impacts protein synthesis, organ development, and function. Epigenetics is the way in which non-genetic factors like our environment can influence the way our DNA is expressed or read. ACEs and toxic stress can affect which books are read and affect epigenetic changes. External factors can be what we eat, smoke, drink, or even experiences we're exposed to. So if we think of our DNA as containing the underlying code for every structure and function in our body, including the stress response, we see that our environment impacts the development of this stress response via epigenetics. In fact, studies have shown how the family environment and early caregiving influence epigenetic regulation of the stress response. In 2008, a study was conducted to compare brains of people who had committed suicide to those who had suddenly died of other factors. They found more epigenetic changes in the DNA extracted from the hippocampus of the brains who had been abused as children and were the victims of suicide. Remember, the hippocampus helps in the stress response and is related to learning and memory. One of the main lessons we take from the study of epigenetics is simply lick your pups. One study showed that pups, or baby rats, who received greater nurturing and licking from their mother grew to exhibit less anxiety than those pups who experienced less nurturing. The investigators were able to examine the influence of nurturing on the rats' epigenetics and observed the pattern remained even as the rats grew into adulthood. Rats that had low licking and grooming had higher cortisol levels, or stress hormone levels. Rats that had high licking and grooming had lower cortisol levels and lower anxiety. Research showed that the nurturing behavior of a mother rat during the first week of life shapes her pups' epigenetics, while their DNA was the same. The epigenetic pattern that mom establishes in her pups remained even as they grew up to be adults, again highlighting the importance of early nurturing and intervention. Geneticists have also found that epigenetic change can be passed down from parent to child, from one generation to the next. New insights of behavioral epigenetics, traumatic experiences of our past, or our recent ancestors can leave molecular scars adhering to our DNA. Childhood maltreatment and aging. An example of an epigenetic change is evident in cellular aging in children who have been maltreated. Telomere shortening. Our DNA is packaged into bundles called chromosomes, and at the end of each chromosome is a telomere. Telomeres are protective end caps that get shorter and shorter whenever a cell divides, eventually altering the chromosomes, leading to cell death when cells malfunction and can no longer divide. If telomeres shorten rapidly, it will lead to rapid aging, and there's an increased risk for age-related diseases earlier in life. This phenomena of cellular aging is correlated to childhood maltreatment. In a 2010 study, adult participants reporting a history of childhood maltreatment had significantly shorter telomeres than those who did not report a history of maltreatment. Let's take a look at a short PBS video that looks at this idea in more detail.
Imagine coming into the world with a person so like yourself that for a time you don't understand mirrors.
As a child, when I looked in the mirror, I'd say, "That's my sister." And my mother would say, "No, that's your reflection."
It's hardly surprising because you both come from the same egg. You have precisely the same genes. And you're literally clones, better known as identical twins. And yet, it's not uncommon for a twin like Anna Marie to get diagnosed with cancer, while the other, like Clotilde, doesn't. But how can two people so alike be so unalike? Well, these mice may hold a clue. Their DNA is as identical as Anna Marie and Clotilde's, despite the differences in their color and size. The human who studies them is Duke University's Randy Girdle. So Randy, I see here you have skinny mice and fat mice. What have you done in this lab?
Well, these animals are actually genetically identical.
The fat ones and the skinny ones?
That's correct.
This gets even more mysterious when you realize that these identical mice both have a particular gene called agouti. But in the yellow mouse, it stays on all the time, causing obesity. So what accounts for the thin mouse? A tiny chemical tag of carbon and hydrogen called a methyl group has affixed to the agouti gene, shutting it down. Living creatures possess millions of tags like these. Some, like methyl groups, attach to genes directly, inhibiting their function. Other types grab the proteins, called histones, around which genes coil, and tighten or loosen them to control gene expression. Distinct methylation and histone patterns exist in every cell, constituting a sort of second genome, the epigenome.
Epigenetics literally translates into just meaning above the genome. So if you would think, for example, of the genome as being like a computer, the hardware of a computer, the epigenome would be like the software that tells the computer when to work, and how much.
In fact, it's the epigenome that tells our cells what sort of cells they should be. Skin, hair, heart. You see, all these cells have the same genes, but their epigenomes silence the unneeded ones to make cells different from one another. Epigenetic instructions pass on as cells divide, but they're not necessarily permanent. Researchers think they can change. And that brings us to the reason why we're showing you twins. In 2005, they participated in a groundbreaking study in Madrid. Its aim? To show just how identical epigenetically they are or aren't.
Our genes are just part of the story. Something has to regulate these genes. And part of the explanation is epigenetics.
Estella wanted to see if the twins' epigenomes might account for their differences. To find out, he and his team collected cells from 40 pairs of identical twins, aged 3 to 74. Then began the laborious process of dissolving the cells until all that was left were wispy strands of DNA, the master molecule that contains our genes. Next, researchers amplified fragments of the DNA until the genes themselves became detectable. Those that had been turned off epigenetically appear as dark pink bands on the gel. Now, the genes from a pair of twins are cut out and overlapped. The results are far from subtle. especially when you compare the epigenomes of two sets of twins that differ in age. Here on the left is the overlapped DNA of 6-year-old Javier and Carlos. The yellow indicates where their gene expression is identical. On the right is the DNA of 66-year-old Anna Marie and Clotilde. In contrast to the younger twins, hardly any yellow shines through. Their epigenomes have changed dramatically. The study suggests that as twins age, epigenetic differences accumulate, especially when their lifestyles differ.
One of the main findings of our research is that epigenomes can change in function of what we eat, of what we smoke, of what we drink. And this is one of the key differences between epigenetics and genetics.
Epigenetics is a study of how external factors impact the way in which our DNA sequence is read and expressed. Stress can impact DNA via epigenetic changes, and changes in DNA expression and can be inherited. Stress and maltreatment can cause our telomeres to shorten faster, leading to health problems such as Alzheimer's and cardiovascular disease. This concludes our module on epigenetics. Center for Youth Wellness, where health begins with hope.